Okay, I'm starting this talk today. It's GlyCosmos and an integrated semantic knowledge base for glycoscience. Okay, first, it's 101 of glycobiology. So I don't think many of you are familiar with the glycoscience and the glycan. So let's begin this kind of very basic knowledge. Okay, so glycan is a molecule in which monosaccharides are linked together by glycoside bonds. So this is this one is a kind of a monosaccharide. So monosaccharides are chained with glycosidic bonds, and those kind of chains called glycan. And glycan attached to a protein. So as here, more than 70% of proteins undergo glycosylation. So this means that all of the protein in our body has 70% of the protein has glycoprotein. And also, do you know that this image is a cell, and glycan can be synthesized in this organelle. The number 5 is ER. It's called endoplasmic reticulum. And do you know where the Golgi apparatus? Do you know do you have any answer to this question? Which is the Golgi? Yes. And number 3? No. Yeah. The answer is number 6. The 6 is the Golgi. This is the Golgi apparatus. So there are two organelles here. So in these organelles, glycan synthesized by enzymes. Okay. And also there are many characteristics of the glycans here. Glycans are very important stuff in our body. So they make disease, and they make some foldings, and they make some equilibrium in the body, so to make our body healthier. And also there's also many stuff you know. This is a protein image of the SARS-CoV-2 spike. And here, this white one is amino acids, and those bushy stuff is all of them are glycans. So they're very important to get some SARS-CoV-2, or when we get COVID-19, the glycan played very important roles. So this is the very important stuff of the glycan role here. And so background. So there are many characteristics, and the important stuff is there is no templated structure in the glycan. So at least we haven't known how to synthesize from any templates for the glycans. So if you get the protein, there are the DNA sequences. So you can get the sequences of a DNA code, so you can imagine what kind of proteins are synthesized or expressed from the DNA coding. But there's such kind of stuff in the glycans. So this is very difficult point when you are trying to get the function of the glycans. And now there are experiments, technology like mass spectrum and also NMR. These kind of stuff already expanded or advanced very rapidly recently, but it takes time consuming to get the function and the structure of the glycans. So above all, the most information found only within the literature. So this is very difficult to share knowledge of structure and the functions as glycans. So here comes the GlyCosmos. So GlyCosmos is a comprehensive web resources for glycoscience research. So there are many databases consisted by GlyCosmos. So there are glycan structures, genes, proteins, pathways, disease, and distribution of glycan in tissues. Those databases are in the GlyCosmos portal site. So you can search for anything related to this kind of stuff in terms of glycans. Yes. So introduction platform for glycoscience research consists of this kind of stuff. So mainly the GlyCosmos has a repository and also tools related to the glycans. And the repository is a kind of glytocan. Glytocan is a repository of the glycostructures. So it's kind of a gene bank in the DNA sequences. So when you get some structure by using an experiment to get the structure of the glycan, you can deposit the structure into the glytoken. And the glytoken gives you your accession number by works. So works is also a data format to represent the structure of the glycans. So glytoken and works is a very important part of the GlyCosmos databases. Yes. And also, the GlyCosmos portal site is built on the semantic web technology. So that's the reason why I submitted this to this SPARQL. So all of the database in the GlyCosmos built on the RDF and the semantic web technologies. So there are many ontologies already we have developed. Here the glyco RDF and glycoco, these kind of stuff are used for organizing glycan databases. And in addition to these ontologies, we also use that another external ontologies to organize glycan data. Yes. And this is a unified ontology to organize all of the database in GlyCosmos to facilitate some functionality for searching the information. So by using this ontology, you can get data by a very fast way, because even though there are many ontologies, but if you want to get any specific data, you have to check many ontologies. But by using one unified ontology, you can get your specified data very easily. Okay. And so this is the endpoints and the integration of the data set. So the GlyCosmos has a SPARQL endpoint, so you can search for any data by using this endpoint. In addition to the web search engine, and also you can use many web interfaces to get information. So if you don't know well about the SPARQL query, or you are not familiar with the SPARQL query or semantic web, you can use a web search engine for the GlyCosmos. But also, if you want specific data by using a SPARQL query, you can get such kind of very good, complicated information by using SPARQL. And here is many databases to organize GlyCosmos. So we don't not only use GlyCosmos databases, and also we can use external database like this one. So thank you for this kind of stuff. And we miss this PubChem. Yes, and so when you try to get some information by using a GlyCosmos, here's a web interface. So you can input your keyword here, and so get, yes. And then you can get these kind of result by putting into the keyword into the web page. So here, for example, the share this transfer the activity, and you can get this list by putting clicking this link. And not only you can get this information from the genes, but also you can get other disease or glycan information by using this search. And so here's a kind of integration, the hierarchy of gene ontology. So this is the ontology you can get by using this hierarchical structure. So we can use the inference functionality by using a virtuous function of the inference. So not only for this specific gene ontology term, but you can get the higher level of the concepts by using this virtuous function of the inference. And also this is a kind of GlyCosmos lectin interface. So you can get lectin information by using that or the similar functionality of the genes or disease. So you can not only get the lectin, but also you can get the binding glycan information. And also here is a pathway information. So this is a localization. So this is a membrane, and it's a nuclei, and where each of the protein or glycan resides in the cell. So you can get this kind of information visually. And also this is an example of the SPARQL query. So it is too small, so this is an example of the result. So this is a disease name in a certain species and the corresponding proteins. So these information cannot only, you can get by using a SPARQL query, but not get by web page. So if you want to get this kind of specific query, you need to get a Spark. You need to use a SPARQL. But and also this is a life science value. So assuming to support various applications in glycoscience by using a GlyCosmos. So our purpose is to provide these kind of aspects in the life science domain. So glycan structure analysis and biomarker discovery, drug target identification, and glyc engineering. These kind of stuff we hope the researchers using GlyCosmos to get the knowledge. And this is the advantages and the future directions. So one is improved data interoperability. So so far, we already get many ontologies and related database on within and also external database, but also there are many related database over there, like Javan told me before that there are repeats and also something, those kind of data are not yet connected or linked to the GlyCosmos. So we want to get collaborations and share knowledge how to link GlyCosmos information, and also enhanced knowledge discovery. So even now we can get some good inference output, but maybe there are room for us to think about what a good example of using a reference by using a semantic web technology. So we are trying to research more based on our knowledge now. And also scalability stuff. So currently, the number of triples are increasing, so we have to cope with a huge amount of triples. So we are now surveying what's the best way to providing information by using a semantic web technology. And also the support for AI and machine learning, and this is the in-progress stuff. So like, as I said before, the enhancement of reasoning capabilities, and also development of ontology defining specific details. And also we are trying to develop tools for researchers to manipulate the knowledge of the glycans, the latest outcome from the literature. In conclusion, efficiency of data integration and accessibility and collaboration. Okay. Thank you very much.
I saw a lot of English on the screen. Do you also support Japanese?
Yes, yeah.
Yes, okay.
Japanese also interface, yes.